Today we will discuss about the LAC OPERON. So, after this video, you should know the structure, function and working of LAC OPERON.
Before we discuss about the LAC OPERON, first we should know what is the gene expression. Gene expression is the process by which information from a gene is used in the synthesis of a functional gene product. These products are often protein, but in non-protein coding gene such as transfer is a functional RNA. Transcription and translation occur during the synthesis of a receptor protein in a LAC OPERON. What is the LAC OPERON?
Word LAC means lactose, while word OPERON means a group of two or more genes having related function. LAC OPERON is a group of structural genes which function as a single transcribing unit under the control of single promoter that involves in lactose metabolism. This process happened in prokaryotes. For example, LAC OPERON is an operon required for transport and metabolism in E. coli.
The main food for E. coli is glucose, and it is used for further activities such as metabolism. However, lactose is a disaccharide which is formed from the condensation of glucose and galactose. Hence, beta-galactosidase is needed to hydrolyze lactose into glucose and galactose. Beta-galactosidase can be obtained through LAC OPERON.
Let us talk about the structure and function of the LAC OPERON. The LAC OPERON consists of regulatory gene, which is also known as LAC-I, promoter gene, operator gene, and also structural genes, which consists of LAC-Z, LAC-Y, and also LAC-A. The regulatory gene, which is also known as LAC-I, is encoded for the repressor protein. And next, the promoter gene acts as an attachment site for RNA polymerase, while the operator gene acts as an attachment site for the repressor protein. Next, the structural genes LAC-Z encode for beta-galactosidase, which is used to hydrolyze lactose into galactose and glucose. And next, LAC-Y encode for permease, which allows lactose to enter the cell by making cell more permeable to lactose. And also last is LAC-A. LAC-A encode for transacetylase, and transacetylase transfer an acetyl group from acetyl-CoA to beta-galactosidase.
So, how LAC OPERON works? LAC OPERON will functions in two ways, which is absence of lactose and presence of lactose. In the presence of lactose, LAC-I is transcribed and translated into repressor protein. Lactose will undergo isomerizations to form allolactose, which is an isomer for lactose. Then, allolactose will bind to the repressor protein. Repressor protein change its conformation shape and cannot bind to the operator. The operon is active. The RNA polymerase will bind to the promoter gene. The translations of genes take place to produce mRNA. The translations of mRNA occur. Then the enzymes, which is beta-galactosidase, permease, and transacetylase will produce. The presence of beta-galactosidase will make the lactose can be hydrolyzed into galactose and glucose. Absence of lactose. In the absence of lactose, LAC is transcribed and translated into repressor protein. If the lactose is absent, the repressor protein will attach to the operator gene and block part of promoter, causes the RNA polymerase cannot bind to the promoter. The operon is said to be not active. mRNA cannot be produced. Thus, there is no transcriptions occur. Beta-galactosidase, permease, and transacetylase cannot be produced. So, for your extra information, you should know that it is better to have one promoter for 3 genes because 3 enzymes can be produced together.
In conclusion, the LAC operon is a group of structural genes which function as a single transcripting unit under the control of single promoter in lactose metabolism. So, the presence of lactose will determine the presence of allolactose, which will later bind to the repressor protein. And the repressor protein will affect the binding of RNA polymerase to the promoter to synthesize enzyme. As the operator is the binding site for the repressor protein. Before this video ends, let's try this question. You may pause the video and try to answer these questions to test your understanding on this topic. The first question is, what is meant by LAC operon? The second question is, state the function for operator gene and promoter gene. And the final question is, what is the effect of permease deficiency in lactose metabolism? You may check your answers, and thank you very much for your attention. We hope that you have understand this topic.